Hi, everyone. Good afternoon. Today, I'm really honored to introduce our most exciting seminar speaker, Dr. Ana Fernandez-Sesma from Mount Sinai. I'll try to keep it short, although I'm too excited and there are too many things I want to mention, so bear with me. Ana got her bachelor's from University of Salamanca, Spain. She then did her PhD and postdoc work with Dr. Thomas Moran from Mount Sinai. And Ana is really a powerhouse in the innate immunity and arbovirus field. She's well known for her work on modulation of immune responses by emerging and re-emerging viruses such as dengue virus, chikungunya virus, and flu. That I'm sure you'll hear a lot of that exciting work today. She's made many important contributions to the field, including how RNA viruses can trigger one of our favorite sensing pathway, the cGAS stain DNA sensing pathway through mitochondrial DNA leakage during infection. And I think she would tell you more about the viral proteins that her group has identified throughout the years that specifically target these proteins to antagonize type 1 interferon response. Ana has a really impressive funding record. And one of a few things that really caught my eye is she was ranked within the top 6 NIH-funded investigator in microbiology in the US between 2015 to 2020. And for the past 7 years, she's been a principal investigator on this very impressive collaborative project, actually also involving Dr. Elaine Reed, our very own, as part of the NIH efforts, Human Immunology Project Consortia called HIPSI, I like the name for it, to really understand human immune responses to dengue virus after infection and vaccination. I'm sure many of you know that it's a very important problem in the world. Some of the really just cool things I want to highlight, part of many honors and awards she has. In 2021, she was elected fellow of the American Society of Microbiology. Actually, this past year, and she just got the award, I went to the award ceremony back in July. She was named one of the 100 most influential women in Spain by Forbes Spain magazine. She's definitely a celebrity. And there were only 3 scientists in the field. And guess what? The King of Spain actually presented the award. So it was a great honor, and it's just awesome publicity for women scientists in general. And finally, Ana cares a lot about graduate education mentoring. She has been the chair, co-director of the graduate program for microbiology at Mount Sinai for 10 years. And recently, our most exciting development for Ana is that she has become the first woman chair of microbiology at Mount Sinai. And she's already making an impact on multiple fronts, including recruitment and promotion of women and underrepresented minority faculty. So, Ana, we're so happy to have you today and look forward to your talk.
Okay. Thank you so much, Melody. Oh, I have to share. I guess I have to share my presentation first. Do I have to close this first?
If we just go here on to maybe.
I leave the experts. So anyway, I'm going to thank Melody for this great introduction. And it's a great honor for me to be here. I guess I'm now a science influencer, something like that. No, but it's really great, not for like me being recognized. I think for me, the important thing is that science is now being considered as part of society, and scientists are actually now making these lists of whatever, that it's not really the award what matters, it's just the recognition of scientists in general, you know, and women in science. So for me, it's a great honor to be able to spread the word, to really kind of educate or like inform about science, and vaccination in particular is a really important thing that we learned from the pandemic. So it's really been a fantastic, you know, experience for me to really be able to, you know, be supported to actually get to these positions and to really be able to spread the word. So I am in Mount Sinai. This is a view of the reservoir in Central Park where I work in the morning. So you have LA, but I have New York. So I'm really, you know, it's not bad. So our lab, as Melody showed, I do it from here, no? So we've been working on dengue and other viruses. I'm going to really tell you the story of dengue because I like to tell a story in which we do like the basic fundamental science and look at mechanisms. And then that becomes something that we can apply in the clinic. So it's really like kind of how what you actually do in your lab with like fundamental science. You always think, oh, who cares? Who cares about this band in a Western blot? It's actually important to see the band in the Western blot. And I get excited about a band in the Western blot, but I also get excited about flow and about other things. So it's really like everything counts and you really build a complete story of what happens. But everything that you do in the lab, of course, you need to validate eventually. So I just want to talk about dengue. I'm sure that most of you are familiar with dengue, but it's an arbovirus, which is arthropod-transmitted viruses, but this is transmitted by Aedes aegypti and Aedes albopictus. And nearly half of the world is at risk of dengue infection. And this is just the areas that we know are affected by dengue. So I guess I have to use this mouse, no? Yeah. So it's mostly tropical and subtropical areas of the world. And this is based on like 2010. But we now know that an estimated 50 and 100 million people get infected per year and is in these areas of the world, but actually it has become endemic in more than 120 countries. And this used to be very impressive before COVID, because over 20 years, we went from like 3 or 4 countries to like 120. Now with COVID, of course, we know that this can happen, but this is really showing how like things can spread very quickly and can become a problem very quickly. So it has this dengue fever that is self-resolving, but also dengue hemorrhagic fever and dengue shock syndrome, which are the most severe manifestations of dengue. So then one of the things that happened is that in the 70s, there are 4 serotypes, which is the most problematic issue with dengue, that they are co-circulating at the same time. So dengue, in the 70s, in Southeast Asia, they had the 4 serotypes, dengue 1, 2, 3, and 4. But then in Africa or in the Americas, there are only 2 serotypes. But by 2010, we had all 4 serotypes all over the place. So this is becoming the issue to develop dengue vaccines. Of course, you probably can imagine that we have to have vaccines that induce seroconversion to all 4 serotypes, and then they cannot wane because there is a problem with these secondary infections. So if you have been infected with one serotype, and the second infection with a different serotype, you're more prone to have severe dengue because of antibody enhancement of infection. So this is actually the biggest problem for the vaccine development. You don't want to make people seropositive and they'd be more actually susceptible to severe disease. So the other thing that actually hampered the research in dengue, first of all, it was exclusively in primates for I guess, thousands of years. But then this is the sylvatic cycle between primates and mosquitoes, but at some point it jumped into humans. And then it actually made its own cycle between humans and mosquitoes. And there is no human-to-human transmission. So we don't have at least documented cases of human-to-human that are efficient transmission. So that also made it difficult to study dengue because you need to understand the contribution of the vector, the contribution of these other factors that might actually make it difficult to study these transmission studies. And the other thing that is a problem is that there is no good animal model to study this disease. So like human primates, like dengue, rhesus macaques, they can be infected with dengue, but they don't develop hemorrhagic fever or dengue shock syndrome. I think it's probably because of a numbers game. You need to infect hundreds of macaques maybe to see these severe manifestations, but nevertheless, then it's not a really good model to study. And also many labs cannot really afford this animal model. Mouse models have been tried for decades. Immune-competent mice, they can show very minimal viremia only with mouse-adapted strains, but they also don't even show symptoms of dengue. They just become paralyzed. So it's really not a good model for dengue. And then the AG129s, interferon alpha, beta, and gamma receptor knockout mice have been used extensively in the field. They actually show high viremia, but they are immunodeficient. In my lab really looks at this interplay between the viruses and innate immune system. So for my lab, this is not a good model because I cannot really see the switches that the virus has to turn off in order to change that active immunity. So some people have used humanized mice, but they just show low to moderate viremia. And also the B cell maintenance is really deficient. So just all this is just to tell you, I don't work with mice. So So the other thing that is interesting about dengue is a positive strand RNA virus. And I was told my famous quote is that I'm too positive to work on negative strong RNA viruses. I was trained in flu my whole career, but I really think positive strong RNA viruses are very unique. They're very interesting. Actually, they have this genome that is quite small, 11 kb, in which they have this even calls one single polyprotein. So it has to be translated in as a one polyprotein. And then they have three structural proteins that are part of the virion, the E protein, preem, and capsid. And this is where we actually raise most of the antibodies against. But then you have all these non-structural proteins that to me, they're most interesting because they really have functions during infection in the infected cell, and they actually modulate many things, including immune responses. So this is the way I kind of vision dengue polyprotein. So I like the way that actually is being described, that it wraps itself in and around the ER membrane. So they are like all these cooperated efforts by the different proteases. So viral protease will mediate the cleavage of all these proteins that will be in the cytoplasmic side and actually cleaves itself. So the NS2B and NS3 make the protease complex. So they cleave themselves on this polyprotein and they actually mediate all these other cleavages. Then you have the host signalase that is in pink that mediates the cleavage of the proteins that are in the luminal side, and they release them from the polyprotein. And then this unknown signalase that liberates the NS1, but also like furin, the infamous furin that is actually now like participating in the maturation of the virion in the Golgi by actually cleaving PR. So there is this almost a choreography of proteases that have to happen for these proteins to be released from the polyprotein. And we know a lot about these different proteins. And the most important thing here is highlighted in red. This is a small virus. It has only 10 proteins. It dedicates at least 4 proteins just to block type 1 interferon. So this is an acute virus, causes acute infection. So these viruses, and all viruses, need to block interferon, but acute viruses, if they don't really deal with the innate immune response, they just cannot establish infection. So this is what they dedicate most of the real estate to, because it's really important to really master this, So I'm going to talk to you about the NS2B3, which is the protease. It's my favorite protein on dengue because it has many, many functions, including interferon antagonism. So this is how we really look at dengue. The infection happens by a mosquito bite in the skin, and then the virus will actually infect dendritic cells in the skin, Langerhans cells, that will get activated and will just travel to the lymph nodes when there is an amplification of this replication. So more cells will be susceptible to infection. So it targets mostly myeloid cells, and then eventually makes it to the bloodstream where it can be picked up by another mosquito. So what we use, the work I'm going to show you, most of the work is using monocyte-derived dendritic cells, which actually could represent some of the different stages of the infection, in which we can really understand how the virus is really manipulating the immune response. So dendritic cells are also my favorite cells in the body because they do have this capacity of like sensing or recognizing viruses or elements that are from viruses. So they have these pattern recognition receptors. And what happens when they encounter viruses or are infected by viruses, even if it's abortive infection, they go through this process called maturation or activation, and they change morphology. They start actually producing cytokines, so they actually are an important part of the innate immune response, including type 1 interferon, which is the most antiviral cytokine, and it actually acts intracellularly. And they actually release chemokines, upregulate chemokine receptors, so they can migrate to the lymph node. And they are, because they process the viral antigens, they can present to CD4, CD8s. So they really link innate and adaptive immunity. So we're quite intrigued that a virus like dengue virus will target dendritic cells for infection, because you have a virus that is infecting the cell line that actually is specialized to sense viruses. So they have to have a lot of good tricks to really manipulate the ability of these cells to detect virus infection and start the immune response. But at the same time, it's a very attractive cell for a virus to infect, because if you can knock down this functionality of the dendritic cells, you can delay or actually block completely the induction of immune responses. So dengue infection in particular is an immune pathology. So your immune response is what's causing the symptoms. So for me, that was especially interesting, because if we understand what happens in the initial moments of the virus infecting the cell, we might understand why the consequences of this interaction with the dendritic cells would have consequences in the adaptive immune response. So I'm not going to show you all this data, but we actually demonstrated that dengue infects human blood and different cells in human blood, including dendritic cells and monocytes. And they seem activated. They produce inflammatory cytokines and chemokines. Then we also found that there is really this lack of type 1 interferon production in those infected cells. So they really including plasmacytoid disease, which are professional interferon producing cells. So the virus actually activates these cells, but actually is turning off the production of interferon. And when we actually compare dendritic cells infected with dengue, we actually try to stimulate them with TLR, RLR ligands, and they actually cannot respond anymore to any of those ligands. And if you co-culture them with naive T cells, they do not induce a Th1 response in those cells. So they actually become non-functional when they're supposed to do, at least in antiviral response. So we found then that what was the protein responsible for this in the virus, and we found that a proteolytically active NS2B3, which is the protease, was required for this inhibition of interferon. So that was really lucky for us, because if you have a viral protein that has an enzymatic function that is a protease, it's probably going to cleave some host factor. So we wanted to know which host factor was. And this was the work from Sebastián Aguirre. That was one of my postdocs, and he was amazing. But he was obsessed with this RNA sensing, but then he was proved wrong. But I told him, follow the virus. The virus is going to tell you where you need to go. But he kept going to RIGI, but it wasn't RIGI. So just to remind you, like RNA viruses the RNA is sensed by RIGI, MDA5, and through MAVs, it actually activates NF-kappa B and IRF3, IRF7. And DNA viruses are normally sensed through CGAS. At that time, it was not even discovered. But we knew that STING was actually an important player, because not only is sitting in the ER membrane, it's also signaling through, is reacting to this DNA sensing, and actually activating also NF-kappa B, TBK1, and IRF3 to produce interferon and inflammatory cytokines. But it also is being shown to crosstalk between DNA sensing and RNA sensing. So at that time, we thought that was kind of the function. So when Sebastián actually he tested all the proteins in the RNA pathway. None of them was cleaved by the protease, although they had the putative cleavage site, but none of them were cleaved in overexpression experiments. So what he did is actually he put STING as a control. That shows that controls are important, because STING was sitting on the ER membrane. And when he actually did is he put human STING, the protease, and then an inactive protease. And we found this cleavage product only when he put the active protease and not the inactive protease. And then the other thing that was quite interesting is that the mouse STING was not cleaved by dengue. So we believe this probably is one of the strong restriction factors for dengue in the mouse, because it cannot deal with this innate immune factor. So we thought, okay, if the virus is actually getting rid of STING, now we knew that the CGAS was discovered. CGAS is really the sensor, and it can send DNA from DNA viruses, from protozoa, bacteria. Actually, any DNA in the cytoplasm will be sensed by CGAS, regardless of identity. So there is really no specific feature, like in RNA, RIGI and MDA5 are super specific for like triphosphates, and like in DNA, you are not supposed to have DNA in your cytoplasm. So any DNA is actually a danger signal. So this is really a danger signal. So then it generates these CGAMPs products based on this DNA sensing, and then STING is really that molecule that continues with the signaling, and it actually activates TBK1, IKK, and then it results in production of interferon. So we thought, what happens to CGAS during dengue infection? And this is actually the previous experiments were overexpression. They are really great because you can do them in 293s that don't express anything. They're really from Mars. You can express anything you want. They don't know endogenous protein, but you need to show what happens in real world, in the context of infection in a primary cell. So we went to dendritic cells, monocyte-derived disease, and we actually infect them with MOG, dengue 2, dengue 4. So we tried 2 of the serotypes, the more distant ones, and then UV inactivated dengue. And we actually found that STING was degraded by dengue in dengue 2 infection, dengue 4 infection, but not UV inactivated. So you needed replication. And this is just the expression of the viral proteins. But then when we went to CGAS, we see that actually CGAS is also degraded in dengue 2 and dengue 4 infection. And actually as early as 6 hours, we see almost complete degradation of CGAS, endogenous CGAS. So that was quite intriguing, because now it really is telling us it's not because STING is cross-talking between RNA or DNA sensing. This is something to do with DNA sensing. So, just to show you that the protease, again, is like a Swiss Army knife. You have one function of the NS3, which is a helicase. It's important for replication, important for many functions of the virus. NS2B was only known to be the cofactor for this protease. So unless they are together, there is no protease function. So they need to come together. So what we did, actually, my animation didn't work here, sorry. But anyway, the surprise to us was that when we expressed them individually, we found that NS2B, which was the cofactor, was the one that when we expressed it, there was a complete disappearance of cGAS. So it was really not a cleavage by the protease. It's not that the protease cleaved, like in the case of STING. We saw this degradation is a different mechanism. So what we did is express NS2B together with cGAS flag in these N293 cells. And we just block all these degradation pathways that we have in the cell for different mechanisms of proteins. So what we look is that they have cGAS, but in dengue infection it disappears, and we only see the reappearance of cGAS if we block the autophagy or lysosomal degradation pathways. So actually, this actually told us that there is a different mechanism of degradation of cGAS by just bringing it to this autophagy and lysosomal degradation. And here I just show you, this is how the autophagy pathway works, and you can block it at different steps. So what we did is block it at very, very early by using 3M. And here is in the context of infection, like before it was overexpression. So we have dengue infection in cells that express cGAS, and then when we actually add 3MA, we could see that this cGAS reappears. So when you infect, there's degradation of cGAS, and then it reappears when you actually block the autophagy pathway. So it's really demonstrating that there is this autophagy pathway, and that this is just a quantification. So it really showed that the virus really wants to destroy the cGAS pathway. So why is this pathway even activated during infection? So the trigger of cGAS activation, we're not sure what it could be. So we went and again to our 293s expressing cGAS. We didn't have very good antibodies to cGAS at that time. So it's cGAS flag. So we had more dengue infected, and we did pull downs, and we decided to sequence everything that was bound to cGAS in infection, in infected cells, and then see if we could identify this DNA. So here we send it to the bioanalyzer, and we really saw very, very nice bands that were enriched in dengue infection. We sent it to our collaborators, which thought that this was crap. And then we actually thought that we really wanted to know what that crap was, so that we were very intrigued. But Sebastian actually started to do PCR and say, let me just try different RNA, different DNAs that could be bound. So he tried genomic DNAs. There was no real pattern between dengue or mock infected cells of these DNAs that were bound to cGAS. But when he went to mitochondrial DNA, in every case, he saw an enrichment of mitochondrial DNA bound to cGAS in the infected cells and not in the mock. So basically, there was a leakage of this mitochondrial DNA during dengue infection that was triggering the pathway, and that's why the virus now has to target both cGAS and STING. And here is just when we got back the data from the deep sequencing, it confirmed again that it was really mitochondrial DNA. So this was quite exciting because this is really telling you that regardless of the genetic material of the virus, the cell is going to be able to sense the infection in a different way. So here is just, this is the work from Tonton, that is, he graduated already from the lab, but he basically was looking at this presence of this leaked DNA in the cytoplasm. So here both dengue 2, dengue 4 induce this leakage of DNA into the cytoplasm that is different from DAPI. So it's really not nuclear DNA. It's really outside of the nucleus. And then he actually, if you look together, they actually are together with the dengue proteins and cGAS. So what we went back to the literature, and we actually found that there is a dengue protein that had been shown to form pores in the mitochondria. So we think some of these viral proteins might actually be on the mitochondrial membrane, maybe to extract some energy or to whatever they need to get from the mitochondria, but that actually results in these pores that might leak this DNA that is sensed by cGAS, and then the cGAMs are generated, and then STING can actually induce the signaling pathway. This also shows why we believe that the virus has evolved to hide the RNA from recognition by the RNA sensor. So the virus makes these membranes, these actually like replication membranes, these vesicles basically, that they're hiding the RNA. So actually this is probably preceded the cGAS pathway, because then the cell actually now sense this perturbation in the ER membrane maybe are causing this leakage of DNA. But the other thing that was quite exciting to us is like, why does the virus really target both? If by targeting STING could actually block the pathway altogether. But it was also a report that shows that the CGAMs that are generated by CGAS could travel from cell to cell. So maybe this is a way of avoiding this signal to be like sent to another cell. But that's kind of speculation on our part. And then we also found that also NS4A, so other proteins have actually been shown to be interacting with the mitochondria. So we then went and said, is this something that happens to other viruses? So we were interested in chikungunya virus, and chikungunya virus is not a flavivirus like dengue. It's an alpha virus, but it's also positive strand RNA virus. It has a completely different genomic organization and replication strategy, but it's also similar in size, and it causes acute infection, but also chronic inflammation of the joints. So people that have chikungunya have had problems, chronic problems after chikungunya. So we had an issue because we had to use fibroblast for this. I mean, it also infects dendritic cells. So in chikungunya, we know fibroblasts in the joints are infected, but the dendritic cells spread the virus to the joints. So we wanted to know what happens in these fibroblasts. And we kind of look at the ones that we normally use in the labs, and none of them express either CGAS or STING, even when you stimulate them with interferon. But this HHF1, this is human foreskin fibroblasts that are primary cells that actually express intact CGAS and STING. So we decided to do the experiments there to look at endogenous. So that's kind of important again, because sometimes the cells that we use don't even express the proteins that we are interested in, but they are good for overexpression. So actually, when we look at chikungunya infection, we also saw that CGAS is completely degraded during chikungunya infection. STING is not cleaved, but actually we found, I don't have time to show you, that there is a viral protein that interacts with STING and probably blocks its function. But CGAS is completely degraded even at 4 hours. So we wanted to know again, like in dengue, and what was the mechanism, and it seems like this, if we block the autophagy lysosomal degradation pathway, we still recover again the expression of CGAS. So it's similar mechanism that what we found with dengue degradation of CGAS. And this was done by Gabe, that is now at Pfizer, working on chikungunya vaccine. So he's very excited. Here he was looking for inspiration in Central Park. So, and so we actually found that CGAS and STING get degraded in dengue infection. The dengue inhibits the induction of type 1 interferon through this by blocking this pathway. And the dengue NS2B protein and the chikungunya virus capsid, and I didn't show you that data, but chikungunya capsid is the one that is causing this degradation. And we know that, we suspected that because it happened so early. Capsid is in the virion, and actually we found that by itself, and I think I have a slide after. So degradation of CGAS in an autophagy lysosomal dependent mechanism. So there is this enrichment of mitochondrial DNA bound to CGAS in these infected cells, which could be triggering this pathway. So I like to say that your mother protects you from viruses, really. So at that time, we were really in a moment in which, okay, we had really figured out most of the mechanisms. We know that these viruses can manipulate in immune responses. And we know that in dengue, we have a lot of problems with adaptive immunity. A lot of non-neutralizing antibodies are generated in after infection. And there is this this manifestation of the disease that is this caused by your immune response. So I team up with Eva Harris, and we put together this HIPSI. We call it the HIPSI. I wanted to call it Divas, but Eva didn't want it. Anyway, we call it the HIPSI, and it was really fantastic because Eva has the best cohort of dengue that you can imagine. She has a cohort of more than 2,000 children in Nicaragua that she's following from 2 years old to 14 years old. So she has all the history of infections. She really has, but she has done most of the work with that cohort was done on adaptive immunity, antibody responses, T-cell responses, but nobody was looking at innate immunity. So what we were interested in that, can we identify things that happen very early after infection that could actually determine if a kid gets hospitalized or if it just resolves the infection? So how can we find these markers that could basically be connected to the outcome? So the second project in that grant was looking at vaccination. So we team up with Anna Durbin, Steve Whitehead, that are doing a tetravalent live attenuated dengue vaccine. And we were able, and they also have a human challenge, so I will show you data. And it was really fascinating to me to really look at these very early times in which you have a like voluntary trial of a human challenge in which you can really identify those early time points. And then in my lab, we were doing ex vivo infections using the viruses from EVAs isolated from the kids, and also the vaccine viruses. So we really wanted to validate that with primary cells. And then we had genomics, proteomics. So we had Nevan, Steve Wolinski, Miriam Murad, and Adib were doing all the immune phenotyping. And then we have data analysis and data dissemination. So it was really a fantastic project that lasted for 7 years. And we are still writing the papers. But the thing that I was really interested in, like looking at what happens with the different serotypes. Do they behave differently? Do we have any signature by these serotypes that are important in terms of what, because for example, we know that, and this is just to show you that it's a flavivirus and it's actually, you might recognize yellow fever, a Japanese encephalitis. There are many other mosquito transmitted flaviviruses, but also tick-borne. So we really were working in this, but dengue 1, 2, 3, and 4 they should be considered actually 4 different viruses. But what we knew is that dengue 2 is normally associated with more severe secondary infections. And dengue 4 rarely causes severe infection. So even talking to Eva, I said, I need dengue 4. And she's like, no, we never have dengue 4 in hospitalized kids. I said, that's exactly what I need dengue 4. Because if you don't have any hospitalized kids with dengue 4, but they are always hospitalized with the other serotypes, that can be something interesting about dengue 4. Because why is this serotype still circulating, co-circulating, if it's really not fit, you know? So we actually, this is Rebecca Hamlin. She's a MD PhD. She's now at Stanford, and she's amazing. She also, all my people are amazing. And she actually looked in dendritic cells. How do dengue 2 and dengue 4 behave? So if you can look, this is plaque forming units. So this is virus release from these cells. So dengue 4 peaks very quickly, and then it goes down. But dengue 2 has this dip, and then it starts crawling back up. But at the end of the day, they both probably produce the same titers. You know, they produce the same viremia if it's an infected person. So, but they have very different kinetics in human cells. But when Kevin and another postdoc in the lab tried in mosquito cells that are with intact RNAi, there was absolutely no difference between the two viruses. So there is something about the human immune response that is making these viruses behave differently. And when we look at the cytokines produced by these dengue 2 and dengue 4, regardless of the time or that we look, it always induces a stronger inflammatory response. Here we're looking at TNF, IL-1 beta, including type 1 interferon. So in dengue 4, we see type 1 interferon, we don't see it in dengue 2. So, and then we see, for example, very strong IL-12 response. So there is really something different between dengue 4 and dengue 2. So Gabe again said, like, maybe dengue 4 cannot degrade STING efficiently. So he cloned the proteases of the dengue 2, 3, and 4. And then this is an inactive one, and then the yellow fever one, Zika virus. And actually, dengue 4 not only cleaves, it cleaves better than the other one. So it's not really a defect in cleaving STING. It's actually more efficient, it induces a more efficient cleavage. So this is really not, and then we found that yellow fever doesn't interact with STING, and Zika doesn't even interact with STING. So it's not really a conserved mechanism between these. So when we look in the PBMCs, here we have PBMCs. This is mock dengue 2, dengue 3, dengue 4. We see already a big, strong signature of inflammatory response by dengue 3 and dengue 4, but not dengue 2. So dengue 2 is inducing a very, very muted immune response. So there is something that is blocking a lot of these immune responses in these PBMCs. And that was done by David. But this was CyTOF. At the time, it was super cool to do CyTOF. Now, apparently, nobody does CyTOF. But I really love CyTOF because it just is basically a combination of like mass cytometry and flow cytometry. And it was really like using these heavy metals. You don't have to compensate. You really can do, for clinical sample, it was amazing. Now we are doing aurora flow cytometry and other methods. But, you know, it was really like a very valuable tool, especially for, we needed fewer cells. And you don't need the compensation that to me was maybe introducing bias. So by using the CyTOF, we actually look in these dendritic cells that were infected with dengue 2 or dengue 4. And actually one thing that was obvious immediately is that the dengue 4 infects many more cells than dengue 2. but they are less bright. So dengue 2 infects very few cells, but they are very bright. So there is already, like, probably dengue 2 doesn't need to infect that many, produces more particles. So there's already some difference. But I wanted to know what happens to these cells that are in the same culture that are not positive for the antigen. How do they behave? So using CyTOF, it allows you to look at the role of the bystander cells versus the role of the infected cells, so, or the antigen positive cells. So here we were looking, in green is dengue 2 and in purple is dengue 4 again, but we had bystander or infected cells. So if we look at TNF, in both cases, depending on the time point, but in both cases, both dengue 2, dengue 4, the production of TNF is by the infected cells. The bystander cells are not producing TNF. But then we went to IP10, which is an interferon-inducible chemokine, and we see that in dengue 4, which we have seen interferon production, the bystander cells are producing this IP10. So this is really telling you that the infected cells can control this interferon response very well, but the bystander cells, because there's so many more infected cells there, but they don't have the viral protein to block it, there is a the phenotype is really coming from the bystander cells. So this is really helping us understanding that not always you're going to have the phenotype coming from infected cells. So what happens to the other cells that are surrounding infected cells? They also respond to cues produced by the infected cells. So, this is kind of a nerdy thing, but it was exciting for us. So the conclusions of the serotypes is that we do see differences in the serotypes, and we chose dengue 2, dengue 4 because they had this extreme epidemiological manifestation, and also genetically they are they are the most distant. So we thought, we can at least compare these two. That all dengue serotypes can degrade sting, so it's really other flaviviruses don't do. But then we could use CyTOF and look at this at the single cell level, and then we could see the contribution of the bystander cells, and it's a very valuable tool, CyTOF or similar techniques. So now I'm moving to the vaccine. So one of the other projects that we had is to look at this vaccine. So the NIAID live attenuated tetravalent vaccine was developed by Steve Whitehead, and the trials are done by Anna Dubin at Johns Hopkins, and they are part of our HIPSI, and they actually created a mutation in the 5' UTR. I don't have time to talk about that today, but we're looking also at the contribution of this UTR because they generate these genomic RNAs that could be sensed differently. So we think that also contributes to the innate immune response. But what they had to do in order to have a vaccine that induced the same levels of seroconversion is to have dengue 1 with this delta 30 mutation, dengue 3 with the delta 2, delta 30 and 31 mutations, dengue 4 with the delta 30, but the dengue 2, they could not just get one that was sufficiently attenuated, so they did a chimera with dengue 4, but expressing the structural proteins of dengue 2. So they actually they see in the rhesus macaque, they saw 100% seroconversion for all 4 serotypes, and it worked very well. So they actually tested them, and they are still doing the phase 3B. They were testing them in endemic areas. So this is really nice. You can test it like in people that are naive, but in people that have already seen dengue, that could be the problem. Can you make them more prone to have severe infection? So they actually look at dengue naive and dengue exposed, and they could see that in both cases they had very nice seroconversion, even if they had been exposed to other serotypes, and they didn't see any enhancement of disease. So that was really reassuring. They could actually continue with the trials. So this is just, it was well tolerated, neutralizing antibodies to all, and Instituto Butantan is doing these vaccines in Brazil and Thailand. So one thing that we tested in the lab, Jessica, she just graduated last summer, and she actually took at the individual viruses, and we wanted to see if they're really attenuated. And this is just virus replication of this wild type and dengue vaccine. And in each case, you see that there is an attenuation in replication. The other thing that we look is just by looking by flow cytometry. We see dengue 4 is the one, again, that infects the most cells, but the ones in dashed lines, they're really they are the vaccine viruses. And we could see that there is this attenuation. And here you can also see when we look at % of infected cells. So it's really attenuated in human dendritic cells. And when we look at the immune response, we see again in that dengue 4, and in this case also dengue 1 vaccine, induce more type 1 interferon than dengue 2, that is in pink, and also much more IP10. So again, we see this profile of this in the context of the vaccine viruses. And so we had access to these to these samples because they did this comparison. They just did a tetravalent formulation in which they include all four, and a trivalent formulation that actually took out dengue 2, and they did a challenge with dengue 2. So with the dengue 2, that was eliminated from the formulation because it was not attenuated enough. So what was really kind of super surprising is that they got 100% of protection against viremia with dengue 2, where they did the tetravalent, but only 20% with the trivalent. So they were very surprised that there was no heterotypic immunity whatsoever. So we actually got a hold of those samples, and we decided, so we are now analyzing those samples, but we decided to repeat that in our dendritic cells. So actually, just to show you that when we look at this dengue 2 vaccine prototype that was eliminated, it actually replicates so much more. So it was really not attenuated. And here I show by flow that it was really behaving differently than the vaccine viruses. This is just the dengue 2 vaccine and the dengue 4 vaccine. But then the other thing that was interesting, this dengue 2, the green one, that is the original vaccine, doesn't produce interferon, is not immunogenic here compared to the dengue 2 and the dengue 4, the chimera dengue 2, but also induce IL-10, which the other ones don't induce. So it's really behaving in a very different way. So dengue 2, in the context of vaccine and in the context of virus, was really kind of behaving in a different way. And when we compare trivalent and tetravalent here, like they did in this trial, the tetravalent, we look at MCP1, tetravalent induced much higher levels of MCP1 than the trivalent, same thing with CCR7. So it seems like it's more immunogenic. We see more signs of innate immune response. And actually, to demonstrate that, we made a vaccine kind of fake formulation with 2 components of dengue 4 instead of this chimera to see if that was the contribution of the immunogenicity. And then again, the purple one is more replication. We see even more than the tetravalent, more IP10 with this double dengue 4 and more VIGI activation. So we actually feel and we think is this dengue 4 component, the backbone of dengue 4 is inducing this immunogenicity and is really providing this, the fact that they did a chimera actually made the vaccine a little more immunogenic. And this is again showing you similar data with other chemokines. So just the final conclusions, this vaccine is safe and is actually now going to be licensed by Merck. And all the vaccine strains have a lower replication, but they induce like low viremia, but they actually are protective, and they induce the different dengue. The dengue 4 seems to be the one that induces the most immunogenicity, and we think that having these non-structural proteins in the chimera for dengue 2 is actually induce a good response in the vaccine. So that was just published last year. So our view right now of dengue 4 and dengue 2 is that dengue 4 has this early viral replication, early viremia, early transmission by the mosquito probably. So that results at the expense of a very high innate immune response, but the virus doesn't care because the virus is gone. So it's not going to act against the virus. So that, but it's creating a very protective Th1 in adaptive immunity that results in clearance and less severe disease and more protection. But dengue 2 has late viral replication, stays longer, late viremia, late transmission. So it has to knock down more elements of the innate immune response. So that's actually causing this really different or aberrant adaptive immune response, maybe these more non-neutralizing antibodies produced. So now we're just looking in this new iteration of HIPSI, which I team up with Viviana Simon, that has a great cohort of SARS and flu. So we are really following only vaccination. So we are looking with Florian Kramer at the vaccination after SARS-CoV-2 in the longitudinal cohort. With Adolfo Garcia-Sastre, we're looking at flu vaccinations. And again, with the team of Anna Durbin and now incorporated Andrea Gamarnik, we have actually access to Sputnik vaccine and also dengue samples from the Southern Hemisphere. And we're looking at immune profiling and also genomics. And this is just the way we do it. But for the sake of time, I'm going to leave it here. And then I can answer questions if you have any. Thank you.
Oh, here, I can. Hello. Thanks so much, Anna. This was a wonderful presentation. So you just presented some really intriguing data on the differential effect of these vaccines.
And I think your conclusion was the dengue 4 vaccine looks to be the winner. The question I have is, has anyone gone further to look at what the boost response looks like? And do you know, is there anything known about how long, like how often someone would have to be boosted in this case? Like, what does immunity look like?
Okay.
Long-term persistent immunity. Is there any information or any data
So,
on
I mean,
this?
what we know is that people in endemic countries, once they have been infected by the 4 serotypes, they rarely get infected again. So because they are in endemic countries, they get naturally boosted every year. I think the problem is travelers, because if they had been infected 20 years ago with dengue 2 and they go back to a different country, and then they might not have sufficient levels of antibodies to really be protected. And 90% of the antibodies after dengue infection are non-neutralizing. So that's really like a problem. So this vaccine seems to kind of avoid that problem, because it seems like these antibodies inducing the vaccine are neutralizing. And then the work with Alex Sete and his group, they were doing with the same, some samples from EVA and also other samples from Nicaragua. They look at this, and some other dengue vaccines failed because they were using different platforms like yellow fever, but they were lacking the internal proteins of dengue. So there are no T cell epitopes to the internal proteins of dengue. So this vaccine includes, for the most part, like internal epitopes for all these other proteins of the dengue serotype. So we believe that that's also helping, the antibody response, because the virus cannot block probably the innate immune response, and having 2 components of dengue 4 is more immunogenic. But then also you have representation of all these internal proteins. But it is a matter of like the epidemiology of dengue that is favoring like people being protected. So I think by the time you're 20, you probably are not going to have, unless you have not been exposed to all 4 serotypes.
Understood. And that's why it's so important to study children
Yep.
in this case.
Yeah. And I can have another lecture just about vaccines, but yeah.
I just have one other question, just your crystal ball. We are undergoing dramatic climate change. This is the vector is mosquito. Do you have any thoughts about what might be happening?
I mean, we're
given
going to see
the
this
global, like the climate change, what might be happening to these, these viruses and their.
I mean,
you know,
we are
life
having a
cycle.
reemergence of these viruses. The mosquitoes are now expanding their area where they can actually thrive. So we have like local transmission of dengue in Spain. We have local transmission of dengue in California. We have actually malaria in the south of the United States. This is happening. People might want to deny climate change. It's not a political issue, it's a reality. It is happening, and we are going to see, but even doctors are going to start seeing dengue, malaria, chikungunya. They need to recognize those diseases. So I think this is important as a concept. We need to like anticipate the appearance of these viruses over and over. It will not cause a pandemic because it's not a respiratory viruses, but it's very hard to avoid being bitten by mosquitoes, and you don't want to be sprayed, and you don't want to be like so it's really a problem that is going to become more prevalent. Maybe in more developed countries with air conditioning, people don't live, but these are mosquitoes that actually bite you during the day. Malaria bites you at night, so you can have nets, and it's a much easier to control. But dengue, you have to go to work, you have to do things. So it's really, and it's a problem of big cities. So it is very difficult to control in Delhi. It's very difficult with Rio de Janeiro, Sao Paulo. So it's really a city problem right now. And that's something that we need to address now in other countries that I have never seen it before. Yeah.
Question here. So thank you for that wonderful talk.
Thank you.
Toward the end, you mentioned that dengue 4, the 3' UTR, seems like it has a particularly unique influence with like immunogenicity. When you introduced the structure of dengue, I actually noticed there were a lot of different secondary structures that looked like they were in the 3' UTR region. Do you think that those secondary structures have an influence on that role in the immunogenicity? And maybe if so, like do differences in secondary structures between dengue 2 and dengue 4
Absolutely.
explain some of the things that you saw? Thank you.
Absolutely. Dengue 4 has a natural deletion of 120 nucleotides in the UTR. So we know that it already has completely different secondary structures. And we know that dengue 4 transmits faster to mosquitoes. So we believe, and also these changes in the UTR, the polymerase binds better. So that's why it replicates faster. So everything is connected, really. You cannot isolate one thing. So the virus replicates faster because the UTR is different, but also it might generate different subgenomic RNAs that are less more immunogenic than the ones that are generated by dengue 2. So we are looking, the other side of the coin we're looking at is like really putting in the context of dengue 2, swapping the UTRs of dengue 4 together with Andrea Marnik to really see what is the contribution of the UTR to the immunogenicity that is really generating these pumps that may be more, or blocking reguide. So yeah, so everything is connected. So it's very difficult because these viruses are different viruses. So they have many different things. So I think each virus finds the equilibrium, you know, it's like fit with a combination of things, which is the UTR plus the best NS2B3 or plus, so they are really like all make the virus more fit and survive by different mechanisms.
Thank you.
Why do you pass? Can you tell them Martin works on 3' UTR?
I love that. And Andrea Marnik is, well, she's amazing with that. Yes.
I was just wondering, so, you know, we're thinking about vaccines for, you know, broadly targeting all these different dengue serotypes, but do you know if there are any good antiviral targets for dengue that would, like, it seems like, you know,
Yeah.
targeting the protease would be a good idea. I mean, there's also the obvious target of the.
I mean,
Polymerase.
targeting the viral proteins is a great concept,
Mhm.
but RNA viruses mutate very quickly.
Mhm.
So you will have resistance very quickly. I think targeting these interactions between viral proteins and host factors, it might be a better way of tackling this, because if you target an interaction with, let's say, sting,
Mhm.
that is only expressed maybe during infection or only necessary during infection, you are not really blocking some of the other functions. So they are like the ideal targets, but the other problem with dengue antivirals is that it is, these are countries that people are not, like companies are not going to make money selling antiviral for Thailand or for Brazil. So it's just the lack of incentive or interest to develop these antivirals and the lack of models to actually test them. So that's also a big problem. So we don't have good animal models. We cannot really do the whole validation. So like my lab is now really interested. We are developing these human tonsillar histocultures, like other cultures that actually might be useful in order to look at these antivirals. So we are part of one of these AVID grants, and we're looking at chikungunya inhibitors. There is a macro domain shared by SARS and chikungunya. So we're trying to see if maybe there is an antiviral. So, you know, there are really things that right now they might be more interested if there is more kind of you know, if it affects other countries in which it is important to avoid this. But antivirals are not easy to develop, I think, but also vaccines are not easy to push, especially the dengue vaccine, that it might take years for follow-up, But
Thank
yes,
you.
of course, and the
Yeah.
polymerase is another one,
Thank you.
I think Anna with the questions.
Mhm.
Is the degradation of cellular sensor by capsid chikungunya virus specific, or this also happens during other alpha viruses?
We're going to start doing that right now. Capsid is a very sticky protein, so I was very, very worried when I got the data because I thought it's just sticking to everything, but it seems to be very specific. So it seems to be this degradation is specific to capsid. So we suspect that other alpha viruses might have the same mechanism. So we are really now looking into some but we want to compare old world and new world. We want to just look at this, how conservative, but this is ongoing right now.
If there's time for another question. Sure, go ahead. Any other questions? Oh, yeah. Go ahead. Oh, okay, go on. Thank you. I'm gonna ask the question.
Thank you for such an interesting talk. I have two questions that I think are unrelated, but maybe are. The first is, I was really impressed at what a good job dengue does in eluding the immune system, which made me wonder, how does the human immune system clear dengue? Because some of these tricks in terms of the interferon 1 jamming are similar to human herpes viruses, which cannot be cleared. And my other question is, does the sequence of which you're exposed to the different dengue strains increase or decrease your risk?
Yeah, so the first one, I think, is because it targets myeloid cells. So I think those cells have a short life, and I think you're not targeting an organ. You know, so for example, other flaviviruses end up in the brain, or end up in the liver, or end up, you know, so flaviviruses, they are kind of almost the same, but they just have a final organ that is different. Is either, so dengue is the blood. And because it targets the blood, it's really, okay, it just goes away, basically. It gets destroyed and it gets eliminated. So, but I think you also get a lot of virus in the liver, but it doesn't seem to cause a lot of damage. Although there are some people that think that it causes damage in the liver or it can eventually go to the brain, but it's not the norm. So I think other viruses like West Nile, other viruses like yellow fever, they really target organs that really they cause damage there in a more long term. So the other one, there is like Eva Harris has the best cohort for that, so she knows exactly the order, and there is a specific order. So there are antibodies to one specific serotype will be more enhancing than others. So the order in which kids get the infection really, really determines their fate. You know, if they get hospitalized or they get more severe infection. The problem with dengue hemorrhagic fever is that they can all be in the hospital, but they don't know what actually is the point in which they crash or they don't. So we still don't know what is the cause of this shock syndrome. They start bleeding from, so it's really unknown. So it's, you know, this NS1 protein of dengue opens the tight junctions in the endothelium, and there's this plasma leakage. And in Zika, the NS1, so that was work also by Eva, in NS1 opens in the placenta. So there is this kind of conserved function, but in different organs again, you know, so Zika went to the placenta. So they find some kind of, I guess it's, you know, just they find an organ in which they can thrive. But yes, there is this order. So that's why it makes it so complicated too. So it was important for this tetravalent vaccine to test it in dengue experienced population, and people already have 3 serotypes. You know, so just to make sure that this is not going to produce enhancement of disease. Yeah.
If there are no other questions, I'd like to ask the last question, Anna.
Sure.
I thought it was so interesting that, you know, by the way, I love the model that, you know, you show for dengue 4 versus 2. I thought it was so interesting that even though dengue 4 seems to be very good at triggering the immune response, it's actually better than dengue 2 at cleaving CGAS or sting. I'm just curious, like trying to sort of tie the 2 parts of your talk together. Do you think there's a fine balance or equilibrium between, you know, targeting the CGAS sting pathway, which is very surprising for RNA viruses, and sort of all these other innate and adaptive immune response that the virus want to trigger or not, I guess?
Yeah, I mean, one of the things I'm not sure is so significant about the fact that the protease from NS4 cleaves better is that these are overexpression experiments. So you might actually have different stoichiometry. So it could be real or it could be not real, but it is really an equilibrium. And I think to me, the lesson is like, you cannot think that one mutation in a virus is gonna make it more virulent. I mean, viruses find this combination of factors that make them actually be better at transmission. So it could be an advantage in the mammalian, but not in the mosquito. So the host switching could be affected. So these secondary structures in the UTR are super important for host switching. So we know that they get eliminated in the mosquito because they're target for RNAi, but then they are a problem in mammalian because they are now triggering reguide. So this is really a dance basically between the virus and the thing. One of the things that is interesting, if you look at the quasi-species in dengue, you look in polio and it's like a sphere like this, in dengue is like this. So you have so many variants because these viruses have so many bottlenecks. In the mosquito already have several bottlenecks. Then between the mosquito and the mammalian, they have to function at 37 degrees and at 28 degrees. So we completely different host factors. So these viruses are very versatile. And you only need a less than 1% of representation of one variant that is the one that takes over in a particular stage. So then in the mammalian, you might have a completely different quasi-species than in the mosquito. So I think it's a cooperative effort of all these virus variants in the same quasi-species. Yeah. Okay.